right. Can you see my slides?
Yes, see your slides perfectly.
All right.
You are in presenter mode. Oh, there we go. Perfect.
There we go.
Now we see it.
Welcome everyone to this third in the series of our Cross Bioinformatic Resource Center TIC webinar series. Today, the topic will be the analysis of TIC endosymbionts, and we'll show what you can do at the bacterial and viral BRC, which we call the BV-BRC, B for bacteria, V for viruses. BV-BRC is pretty recent. It's a combination of the bacterial BRC, which is Patrick, or was Patrick, and the viral BRCs, which are Viper and IRD. So this is going to be a kind of whirlwind tour, like all of them are, but these are the ones that we've had in the TIC webinar series. And you can register for those at ViewPath.db, which is our sister BRC, or brother. I don't know why it has to be sister. The URL is at the bottom. The recorded videos are present on both YouTube channels for both resources, or you could get them here. Yep, doesn't seem to want to go down. Today, I'm going to be discussing a really fascinating paper I found on the interactions between ticks and their bacteria. This was done by a group in France at Montpellier. Olivier Durand is the lead author, and it was a really cool analysis on the interactions between ticks and their hosts. And it was more than just pretty pictures, although I'm sure you must all admit that this is like one of the most awesome tick pictures I have seen. So what the authors did was they found out that ticks need a source of vitamin B. Who knew that ticks needed something like that? So they set up this experiment where they treated ticks with antibiotics or withheld antibiotics. When you treat it with antibiotics, that would, of course, kill off any bacteria in the tick. And they treated them with B vitamins if they supplemented them, or withheld supplementation. Now, in most instances, without the antibiotics, the ticks were fine. With the antibiotics, if you gave them B vitamins, the ticks were fine. But this is a picture of an unhappy tick who is on his way to dying because he doesn't have his B vitamins. So they were able to make the association that the bacteria present in the tick are providing it with B vitamins. So not only did they do this great observational paper, but they did a deep bioinformatic analysis of the genomes of these bacteria. So they went in, they isolated the Malpighian tubules of the ticks, which are sort of similar to our kidneys. They isolated the bacteria from that. They sequenced it. They looked at the read quality, and then they did assembly using Abyss and Spades. They did an annotation of the genome that they got. They used BASIS, something I've never used before. They created their own protein families and binned those protein families into functional families that concentrated on metabolism, vitamin and cofactor synthesis, and amino acid and nucleic acid synthesis. They generated a phylogenetic tree using RAXML. They did a genome alignment using MAUVE. They used BLAST. And then they did a deep dive into the biosynthetic pathways for vitamin B and also the virulence factors. So their tree that they produced, they used 425 single copy orthologous genes, and the tree was based on 133, almost 134,000 amino acids. And so they could see that the isolates from ticks, and this was their guy, Francisella F-OM, all clustered together on the tree. And this made me think, because what the one organism's pathogen might be another organism's pal, in this case, the tick. They did MOV genome alignments, looking at rearrangements between the isolate that they had from the tick and another tick endosymbiont, and a vertebrate pathogen and the tick symbiont. And then they compared the pathway genes and the virulence factors. So those are really, this team had deep bioinformatic cred. They put the contigs in GenBank, not the reads, alas, because I wanted to show you an end-to-end analysis that you could do with this data, but we have what we have, and it was good of them to put it here. So we have the contigs. And so the question I wanted to show you all today to bring up is, could you do a similar analysis? Now, I'm just a pretty much straight up biologist. I can't do much coding. So this team, they have their own GitHub repository where they put their scripts in it. They tell you exactly everything that they used. But would a regular straight up biologist be able to do this? And could you do it in the BV-BRC? Well, let's step away from here and go over to the BV-BRC. Oops, let me pull that guy out of the way. And let's look to see if you can do a similar analysis. Now, today, some of the things I'm going to be discussing is showing you briefly some of the data that we have available. We have a number of tutorials and webinars and videos. This is all going to be pretty fast as I try to recreate this experiment, but there are things you can refer to, and a lot of them are about the different pages and stuff. So I don't want to go into a deep dive on the data on every page or how to do the searches or the workspace and the analysis. So I would refer you to those videos. But let me point out that when you're logged in, like I am here with my little very boring avatar icon here that tells me I'm logged in, when you were logged into BVBRC and your Patrick, Viper, or IRD login works as well, you have access to our tools and services, and you also have access to your own private workspace, and you can also look at a collaborative workspace and a public workspace. Everything that I'm going to go through today has been stored in the public workspace, so I'm going to step you through it right now so you can see it. This is the public workspace, and we'll have to scroll down to BV-BRC workshop, and then here to tick webinars. And here's the endosymbiont folder. And the reads that we're going to use today are in the comprehensive genome analysis folder, which has the contigs here. And every single thing that I show you today, I've got the pictures, the Excel files, and the completed jobs. And I've also provided you with the manuscript by Duran et al. In the supplemental data. I'm not sure if that's the right thing to do, but it's all there for you. So let's go back to the BV-BRC. And let's click on the organisms tab. You can see that when you're looking for data in the BV-BRC, we have bacterial pathogens, we have viral families, we have featured viruses, and then we have some direct links to all bacteria, all archaea, all viruses, and eukaryotic hosts. Now, don't be misled by just seeing these genera for the bacterial pathogens. We have all bacteria in BV-BRC. In fact, every couple of days we go into GenBank and bring in new data and annotate it and have it available in the resource for you. So if you work on cyanobacteria, or if you work on something like Buchnera, it's all here for you. So don't think every week somebody says to me, I can't use your resource because it's only on pathogens. It's not. It's on all bacteria. So let's just click on all bacteria here. And then you, once again, it's the pathogens. But what I want to point out is the number of genomes that we have in the resource, which is almost 550,000 genomes. So now you're starting to see, I mean, it's a gold mine, but it's also a problem for you, right? With this amount of data, how do you find things that you want to compare your data to? So I'm going to step you through that today so that you can see, here's my data, what's close to my data, and how can I compare it to the other data in there. If you were interested specifically in Francisella, I could go through the organism tab there, or I could click on this hyperlink here. And that's showing you the taxon landing page for Francisella. And it's telling you that there are over 1,000 genomes here. And this is the taxon landing page. It's got a number of different tabs where we have even more information on that. But let's see, do we have this particular genome in the database? I could look through the genomes, but I want to show you how to use the global search, which is up here at the right, or if you're on the home page, it's here. So I know it was Francisella, and the authors called it FOM. So all data types, I could focus primarily on the genomes. BVBRC speaks genome for the most part, and I could do a more stringent search, but I just want to look at all terms. So I'll click on the search icon. Oh, and look, right away, it pops up. Francisella-like endosymbiont strain F-OM, and it gives me the GenBank accession. It tells me the size, the number of genes, when it was collected, the isolation country, and who it came from. If I click on this checkbox, the vertical green bar becomes populated with possible downstream functions. So I want to, this is the information on the genome, but I could click here on the genome and switch to the genome landing page for that organism. And I just want to be sure it's the right one, right? So like I said, this is the genome landing page. Like the taxa landing page, there are a number of different tabs to allow you to drill into the data in different ways that's of interest to you. But there are other webinars that we have that covers that. Right now, I just want to make sure this is the guy I want. So here's the strain, there's the taxonomy information. These are all links to different repositories, so you could see more information there. Tells you where it was sequenced. And if I scroll down here, isolated from the host Malpighian tubules in this laboratory in France. So I feel this is my genome. This is the one I wanted. And here's all the information about that. So we know that that genome already exists in the database, but let's pretend that it doesn't exist or you didn't know it existed. Could you do the same analysis here at BV-BRC? So let's click on Tools and Services here. And you notice we have a variety of genomics tools, protein tools, metagenomics tools, transcriptomic tools, and different utilities. The authors who described this particular isolate and genome first started with FASTQ utilities. They did a FASTQC, which tells you the quality of your reads. Okay, we don't have the reads, right? Because they didn't put them in the sequence read archive. They put the contigs in GenBank, but we could have done that here. You can start with paired reads, single reads, or get something directly from the sequence read archive. And then using this service, you could look at read quality using FastQC. You could align reads to a specific genome. You could trim the reads, or you could even make sure that your paired reads were equal in number. Sometimes that will cause an assembly service, an assembly job to fail if you have the forward strand reads, or there are a lot more of those than the reverse strand reads. So you could fix that here. So I just wanted to show you that you could do that here at BV-BRC, but we don't have the reads. So let's look back into tools and services. Because we don't have the reads, we also can't do an assembly, but you could do it if we had the reads. The assembly service takes paired reads, it takes single reads, it takes an SRA accession, and it also allows you to do a hybrid assembly. Like if you had PacBio reads and Illumina reads, you could do a hybrid assembly. If you click on the strategy here, hybrid assembly using unicycler or spades. Canoe would be for long reads, metaspades for metagenomic samples, plasmid spades to try to pull a plasmid out. And if you have single cell, you would have that type. But remember, the authors used spades. We have spades too, but we don't have reads, so we can't do the assembly. Where we'll have to start is with the annotation. So we can do the annotation or the comprehensive genome analysis service, which I'm going to show you today just because I like the report that comes with it. So let's click on comprehensive genome analysis service. And you notice that it starts, you can start with reads, which it have the same parameters that I showed you with the assembly, or you could start with assembled contigs. We developed this service because we used to have assembly and annotation, and we still do separate services. But when we worked with biologists and workshops and stuff, we found that it was just, this is more what they wanted. They wanted a unique service, a united service in which they could do everything and it wouldn't be this hard, all the clicking. And they also wanted some indication of giving them more information at the end. So this particular service does that. So we're going to step through it. Contigs, and we're going to go to the workspace and get those contigs. To do that, you click on this folder icon, and this opens a pop-up window that shows me my workspace where I could upload reads to it, and I could create new folders and upload reads to that. But these particular reads are in the public workspace. So I would click there and click on public workspace, and then scroll down, as I showed you earlier, to BV-BRC workshop, and then scroll down to TIC webinar, and then endosymbiont. I know it's a lot of clicking, but it's okay. It's not going to kill anybody. This is a config file. So I click on that and I click OK. And now I've done the first step to start an annotation service. Now I need to fill out some parameters. We allow you to do annotation of bacteria, archaea, or viruses. This is a bacteria. Now we have to specify a taxonomy name. So you could put anything in here. I mean, it has to be a bacterial. You could start with bacteria. Any genome that comes into the resource goes through this pipeline, public or private, and they all have similar things that happen to them in the annotation service. Every genome gets a cross-genus protein family. The set of protein families, we call those our global families. They're Kamer-based protein families. Remember, the authors did that manually. We do it as part of the annotation service, so that you can make comparisons across the genus boundary. If you give the genus or lower name, genus or species name, then you get the local protein families for a more deep dive analysis. So I always encourage people to try to go as deep as they can to try to at least to get to genus. So you get the 2 flavors of protein families. So I started typing that and I click on that, and you see the taxonomy ID there. So I have to give it an identifier, and I'll say endosymbiont April. I've annotated this a couple times when I've been practicing for this. So I'm giving it a unique name. And you notice when I type that, it's showing me what the name's going to exist in the workspace with. And then I need to give it an output folder. I could navigate to that in my workspace, or I know that I've named some and I'm going to put it in my Nobel Prize winning research folder. And then you notice the submit button is blue. So that means I can submit this job. So I'm going to go ahead and do that. When I do that, it tells me that I've submitted the genome analysis job. And now it says it's been submitted successfully. And so I can check my jobs list to find out how it's going. So I can get to my jobs by going up to workspaces and clicking my jobs, or just clicking down here. And you can see that my job it says it's running. Actually, it's probably just queued right now because there are a lot of people that use the service. So we do have a queue. Now I've run this job before. So, and you can see I've run a lot of jobs. So I've told you that it's in the workspace, but I want to show you one way you can find how to access the job from your jobs page. So I'm just going to filter down on mine to the Comprehensive Genome Analysis Service. And then here is the job I ran earlier. So when I click on that, the vertical green bar gives me a number of possibilities. I can view the job, I can report an issue. If you have a job that failed, let us know so we can look into it and see what happened. And I could also rerun the job, but I just want to view that job. Now you can see this is what happens when you run the Comprehensive Genome Analysis Service in BV-BRC. And there are a number of downstream files. The annotation one is really good to look at. This is the completed annotation job, but this is my favorite one, which is the full genome report. But before we get to that, I want to show you what this looks like in BVBRC. So up here, I can do the view icon and the report icon. I could get to the report this way, or I could click on it here. But I want to see the genome that's now been integrated into the website. So I click on that. It rewrites the page and shows me the genome landing page. And remember when I showed you the one that was public in the system? Looks very similar to that, except you don't see the information about where it was isolated and things like that. This is my private genome. The only reason you're seeing it is because I'm sharing my screen with you. If you're logged into your account, you're not going to see my private data. But my private data is here, and it looks, the tabs are the same, the structure is the same, the information is the same, and more importantly, I can look at it throughout and compare it to any of the data, any of the 550,000 genomes in the resource. So that's what it looks like on the page. But I want to show you the report, because this, I think, is the most valuable part of this service and why I would recommend it that people use it. It gives you right up on top an idea of the quality. We found that a lot of researchers who weren't experienced in examining genomes, it was hard for them to determine if their data needed some help and if they needed to do some more sequencing. So right at the top, we tell you if it's good or poor quality by comparing it to other genomes that are closely related to it in the service. We give you details on the assembly. Now, I know we didn't do an assembly on this, but there were contigs on it. And so you can measure this sort of information. So we've done that from what we could get from the contig file. Yeah, there's the taxonomy, information about the genes that are called, information about the proteins. Remember, I told you that every genome, public or private, gets these cross-genus families, protein families that are Kamer-based. They also, because I specified the genus Francisella, I get the local families for a deep dive, and I'm going to show you how you can use those later today. We provide you with this circular graph that shows you the contigs on the outmost, the forward strand, the reverse strand, the ribosomal RNA genes, the antimicrobial resistance genes, virulence factors, GC content, and GC skew. So you may be wondering, what are antimicrobial resistance genes and virulence factors? Every genome that enters our system is blasted against specialty databases, one for antimicrobial resistance genes, for virulence factors, for transporters, and for drug targets. Because we talk to researchers, these are the kind of things that they're looking for in that information. And rather than making them do this blast manually, we're providing that for them. You'll notice that some of these genes are colored. Those colors are based on a functional analysis that we do called the subsystem analysis. So for example, protein processing genes are colored orange, and you can get an idea of where they are in the genome. They're on that particular contig and in this area. The specialty genes are what I were describing earlier, what we did with those special blasts, so that you can look and see which genes might be of interest to you and which had hits to significant hits to those resources. In addition, we also have Kamer-based antimicrobial resistance genes that we have developed. And it also shows you those because people are, of course, very interested in antimicrobial resistance. And finally, we do what I call a quick and dirty tree because it's only based on 5 genes. We take your genome and some of the high quality reference and genomes to compare it with, and we take 5 cross genus families, of which they each have 1 copy per genome, and then we create a quick tree of that, and here is your genome within that tree. So it gives you an indication of where your genome lies in the greater scheme of things, but I wouldn't use this in a publication. And finally, at the bottom, because we want you to publish and we want to make it easy for you, up here we referenced each of these things so that when you write your publication, you can refer to these references. And I was just at a workshop where someone asked me, would it be plagiarism for me just to take text directly out of this? No, go ahead. I want to make it easy for you to publish. So the original authors, Duran et al., they did FastQC, which we could do, but we didn't because we didn't have the reads. They assembled using 2 processes, one of which was spades, which we have, and they did an annotation, which we did. Now you might ask, what genome, what's close to my genome? How do I find out who its closest relatives are? In this, we have a tool for that. So let's click on Tools and Services, and then click on Similar Genome Finder here. Every genome that comes in, we assign, there are 1,000 Kamers that are assigned to it. So, and then we look for a comparison of those Kamers across all the other genomes. So if I was going to look at my private genome, I'd start typing here, and look, this lock at the top tells me that's my private data. It's trying to help you find your data. So we click on that. You could also, at this point, I could have uploaded the FASTA file or even read files to look for that if I wanted to do that earlier. And then we want to look across all the public genomes, and this is telling me who I'm looking at. So I click the search icon, and then sometimes we'll have to wait a few seconds for this to load because it's going across 550,000 genomes and looking at how many shared Kamers all of these things have. So while we wait, I'll have a sip of coffee. Sometimes it's fast, sometimes it's a little bit slow. But that's not too bad. Okay, so the top hit, and notice the green bar, nothing here. When I click the first one, the top hit is my friend from France. And look, it shares 1,000 out of 1,000 Kamers. No surprise, because they're exactly the same thing. Remember, I took the same config file and annotated it, so no surprise that it's the same thing. The next closest hit is this one, which is an endosymbiont isolated from the same strain, but, from the same tick, but it has a slightly different strain name. If I click on that, I want to see who did that study. So that was isolated in Portland in the United States. And so you can find out more information. Well, it wasn't isolated there, it was analyzed there, but it was from the Czech Republic. So each of these, I could find out more information about the ticks that were, you notice here, it's saying that what the host was from these, which were these ticks, and find out more about this one. That one says it's in this was from North America. And then there's this one that the authors used a lot in their analysis, this Francisella persica. And if I scroll down here, it was sequenced in Nebraska, and it was isolated in a heron rookery from a tick. Oh yeah, and they tell you which soft tick it was. So, I mean, that's pretty, so the top ones, this one only shared 167 of the Kmers. This one had 322. This one had 991, and this was a exact match. Of course it would be, because it's the same thing. So notice when I've selected a number of genomes, I could actually create a genome group of these and do things downstream with it, like look at, compare the pathways across them, or use it to build a phylogenetic tree. So to create a group, I'd click the group icon, and this opens a pop-up window. And I would say I want to create a new group, and I will call it Francisella tick genomes. You know, call it whatever you want, but there you go. So that's been added. So one of the things that the authors did is they created this tree. So now I want to create a tree like this. And notice that they provide what every biologist does, and what you would need in a publication anyhow is the genus, species, subspecies, and the strain name for these things. But let me point out that the strain name is not necessarily unique, and so it can create some problems. So I want to try to recreate this tree and see if I can do it in BV-BRC. So I'm going to move this aside, and I want to go to my organisms, and I want to go to Francisella, and then I want to click on the genomes. And remember, well, let's wait for it to load a second. So these are all the genomes, and if I click on these filters here, you can see true genomes and false genomes. False genomes, it means it's my private data. So let's click on the true genomes, and there are a number of things that I could tone down the data, filter down the data to be able to get a specific group. Like if I wanted all the ones that had been isolated from ticks, I could easily do that, especially if they had that information within their GenBank record. But I want to look for these genomes exactly. So I can use this keyword search. And one of the first ones was FFC198. That was one of the genomes. Oh, and look, there it is. Well, that was easy. So I can click on that, and I'm going to create a new group of that, which I'll call Duran Tree Group, just so I know what that's called. And then when I can add that, and it gives me a message that I've added it. So I'm feeling really confident. So another one that they did was SCHU. So I click on that. So I click on, okay, SCHU, and then there was this S4. So I click on that. Oh, and you see, this is why the names are a little bit problematic. in that they're not unique. If they'd given me a GenBank accession, I could work with that because those are unique identifiers. So I would just have to choose one of these. I don't really know. When you look at their tree, there's no indication in their paper exactly which genome they used. So I'm just going to have to guess. And I can add this one to the group and add it to an existing group. And when I click on this drop down box, it's going to show you the one I most recently recently created. So I would click on that, the Duron tree group, and add it. Well, rather than make you guys sit here and watch me click on each of these laboriously, I've already done it for you. So we're going to step through it so you can see how to do the tree service. So let's click on Tools and Services and then Phylogenetic Tree. So I can look at, if I click on this box, it'll show me, or the down arrow, it'll show me the genomes I've most recently grouped together. And really, I want to go back to an earlier one I did, which was the Francisella Duron genomes. And I need to add those to my selected input genome table. And if I wanted to add my private one, I would start typing here, Francisella, and here he is at the top, and add that there. And so you can do it one by one, or you can see how creating a genome group would make things really easy for you. Let me describe the service a little bit and what it does. Like the Duron paper, it looks for single copy orthologous genes, but we unite those in the global or the cross-genus protein families, or the global protein families. So it's looking for PG FAMs that each of these genomes have only one copy of. And then it takes the nucleotide sequence for each of those genes and the amino acid sequence for each of those genes and creates a tree for it. So the output folder, we would want it in my Nobel Prize winning research folder. I need a name, Duron. I run a lot of these things, so it's important to me to have a lot of information in the name so I can figure it out later. Those authors used 425 genes, so I can use 500. Now, let me point out, the higher you go with this, the longer it's going to take. So if you want something fast, 100 genes is probably plenty for a tree. You'll want to, I'm a biologist. I'm like you. I want all the data possible. I'm going to put those 1,000 genes on there, but then I'm going to wait maybe up to 7 days for my tree. So be careful what you wish for. Try to scale it. Start small and then build your way up. Look at your tree. Look how well supported it is. Do you need to add more? You may not. That may be enough. This allows you to add protein families that, let's say you have a really poor quality genome, but you still want to see where it trees out. You can use these kind of things that says, I'll look for protein families where some of the genomes are missing them, or some of the genomes have duplicate genes, but we don't need to do that. And then I could submit the tree job, but I've run this tree a lot, so I don't need to submit it, but I will show you what it looks like. So I'm going to go into workspaces, public workspaces, BV-BRC workshop, tick webinar, endosymbiont, and now we're going into phylogenetic tree. And the first tree I ran was this one, this checkered flag, like NASCAR, or I guess Formula One, means you cross the finish line, this job completed. It can double click on this row and it's going to show me, I could view the tree. There's a lot of information that come with these trees. So I'd encourage you to look at the tutorial so that you can see all the different flavors of it. But it includes things like a list of the genes that were used, a list of the genomes, a list of the data. So all of those things are things you could pull and put directly into a publication. But this is my favorite part of our tree report, is this report.html. So I click on that. I click on the view icon. And here's the tree. Now I'm going to pull this guy over. This is Duran et al.'s tree, and I'm hoping you're going to be able to see both of that. Tularensis, Media statica, Holoarctica, Novacida, Hispanensis here. Here are tick guys right here. And here they are for us here, followed by the Orientalis guys, Phyllomiagra, which is down here, and then all this other stuff. So it's pretty cool. I mean, it looks good. Also, look at the support values. We have similar support values to be able to see that. So that's a well-supported tree. But I want to show you more things about this. If you go down to the tree analysis statistics, it says, oh, you look for 500 genes. Uh-oh, we were only able to find 393. But of that, it included 125,000 aligned amino acids and 376,000 aligned nucleotides. Those are statistics you can put directly into your publication, and that will quiet any reviewer, especially, and you know, the tree method was in RaxML and the bootstrapping was RaxML. Well-known program, the authors used it too. Any reviewer is just going to be quieted by that because that's a lot of information and they're going to trust your tree. But my favorite part, we're going to scroll down. It tells me what genomes were used, what gene family statistics were used, what gene families were used. And that's a lot because I asked for 500 and it got 393. So bear with me while I scroll down. My favorite part of this is at the bottom, it says, oh, look, you asked for 500 and you only got 393. If you get rid of this guy, you're going to get more genes. So who is that guy? If I do a find, I can see, oh, this guy is the whole Arctica FSC 200. I've got a lot of those. It doesn't add anything to the tree. I could get rid of that and make a better tree. Well, I already ran that for you, so we're just going to look at that briefly, really quick. So I'll click on that, and here it is, Francisella minus FFC 200. So I click on that, and then let's look at the HTML. Let's look at that. It's going to look similar. Note that I also have this picture in that folder too, so you could look at that to make that comparison. Yes.
There's a question here.
Yeah.
What is the method being used to assign genes to synteny/orthology groups?
It's the global protein families that are assigned with any annotation. What the algorithm does for the tree building is it gets the group of genomes, and it looks for those global protein families, which we call PG FAMs, that have only one gene in that protein family. So if there were 40 genomes here, there'd only be 40 genes. If it had more than that, we wouldn't use it unless you did those manipulations that I showed you at the beginning, where you could look at protein families with deletions or protein families with duplications. So this one and what it does is it takes the gene sequence, so it takes the nucleotide sequence and the amino acid sequence, and so it concatenates all those together and builds the tree from that. So the Duran paper using RaxML, like this algorithm does as well, the Duran paper just used the aligned amino acids and they had 133,000. We're using nucleotides and amino acids. So when I look at this tree, which is removing the difficult one, there were 500 genes requested. It could only find 424, but look at this. It found 134,000 amino acids, which is very close to what Duran et al. Had as well. And in addition, it had 402,000 nucleotides. So that's a powerful tree. I just love this service. So anyhow, so from the manuscript, reads, assembly, annotation, trees. So far we've been able to do all of that in BVBRC. The next thing they did, yes, question?
Rebecca, can I ask a question? Actually, this is
Yes.
from me, but as I was
Sure.
watching, is there a limit on how many genomes you can add to a group, and also?
Oh, there's no limit on how many genomes you can add to a group, but there is a limit on how many genomes you can add to a tree. Right
Okay.
now, just because you're my closest friends, I'm going to tell you this. Right now it says 100, but actually you can add 200 because there's a little bug and it hasn't been fixed yet. But another thing you have to do is think about it.
Right.
What's going to look good in a publication? There'll be the initial trees I run, but then I'm trimming down because I want to present my reviewers with the best possible image. And if you have even 100 genomes, I think is too much for a good tree, for a publication view, not for your own private information, but for a publication view. So
Right.
anyhow.
And also maybe another follow-up question in terms of the file, the different file formats that you're getting. I notice you also have in your directory, you had, PNGs and a PDF.
Yeah.
And
and you could take those directly.
Okay, and what about, and I see you also have the .phy file. So somebody could potentially take their.
Oh yeah, we have the Newick files, and this is the Newick files with genome IDs. If you click on the detail files, there's even more. So you could even see, not this one. I want to see this one. So you could see, well, that wasn't quite what I wanted, but they're all different things.
But that's really cool because then anybody, if you want to, for example, draw your tree in a different software, if you have your own software or there's another web service that you like the way they draw things better, you can get the Newick format or whatever. That's great.
Right.
Yeah, very nice.
And in the tutorial for this, we talk about how you can take it into Fig Tree, which is one I often use because iTool now has a paywall for it, but Fig Tree is still free. So you can put that in. And also these trees, I should note, are midpoint rooted. So you could, if you want to include that in your publication.
Right. Cool. Thank you.
So now we're going to go into genome alignment. So remember that the authors looked at the two tick things, and they used the Francisella persica tick as the reference strain. So we can do that. And do you see how easy that is? Just to click in there and add it. And then there was my Francisella. We have different ways that you could do this, but it's so easy for me to do this. And then I would put it in my Nobel Prize winning research, output name, Persica v. Indo. But I've already run this, so I'm going to show you what it looks like. But it's that simple. So I'm going to go into public workspace, and I know it's annoying to watch me migrate by here, but suck it up, buttercup. That's the way we're doing it. Tick webinars, endosymbiont, genome alignment. So the checkered flag means it was successful. So I click on that. And all we have is this view right now. We don't have, other than these downloadable files, this view of it. Sometimes it's fast. Come on. This is, remember, it's progressive moth. But I just think it's very pretty. I don't know why it appeals to me so much because it's just boxes, but I think it's the straight corner as opposed to mauve, which has this is progressive mauve. I just like it a lot. So that was the tick versus tick, the public versus my endosymbiont, and it's showing you like this thing was rearranged here, this was maintained in the order in the genome. I also did a similar job for the intracellular vertebrate pathogen versus the endosymbiont, which the authors did. And we can view that here, and it's noisier like theirs was noisier. So we've ticked off protein, we've ticked off trees, and we've ticked off genome alignments. Now, the next thing that the authors did was a deep dive on the pathway analysis. Right now in BV-BRC, we are in the process of migrating, creating a new tool from some tools we currently have at Patrick. So I can't show it to you in BV-BRC, but I can show it to you in Patrick and you can imagine it. This is the comparative pathway viewer. So I'm going to click on that. And our authors, when they did this, and I'll pull this little guy over so you can see, they looked at the 2 tick genomes, the intracellular pathogen, and they compared it to a Coxiella endosymbiont also found from ticks. So we're going to recreate that here. So I need my genome, which was Francisella. There's the endosymbiont. And see how easy this is? I could add a genome group, but I didn't create a genome group from these just because this was just almost as easy as that. The Persica, which was the other endosymbiont. And then it was the SCHU S4. And oh gosh, there's so many of those, but I'll just choose one. I can't remember which one I chose earlier. See, that's what the problem with some of the strain names, as I told you before, they're not specific enough. And then it was Coxiella like CRT. There it is. Add that. And now we click the search here. And what this is going to do is look across all the pathways that it's going to take these genomes, and every genome that comes into the resource, we map them to EC numbers, and EC numbers are enzyme commission numbers that are also part of pathways. So I'm not going to, the authors did an exhaustive review of this. I'm just going to show you, oops, where am I? I want to find the biotin pathway. I just wanted to show you biotin. There are 131 pathways, but biotin is vitamin B7. So I can click on that. And then you notice there are some different icons here, one of which is the map, the pathway map. So I want to click on this. And for those of you who are familiar with Keg, we actually do the Keg Pathway Maps. So this may take a little bit to load, but it's showing you the EC numbers here. And if it's this lime green color, if I click on the legend, the lime green color shows that every genome that I'm looking at, that I put into the analysis, has that present. If it's this olive drab color, it means it's annotated, present in some, but not all genomes. Now you notice from this guy here, they only looked at a few of these genes for biotin B7. In this one, there are a lot more, but to be able to see in more detail what those are, you can click on the heat map view here, and you can even arrange these things in the same order that the authors had the genomes, and I could arrange them in the same order that they appeared in this diagram. And it was a little bit tricky because here they used the gene symbol instead of the name, but in the virulence factor, the virulence factor part of this, which I'm going to go into next, I'll show you that. And at the end, I'll show you exactly how these things lined up. So we've done pathways, we've done genome alignments, we've done protein, we've done phylogenetic trees.
Rebecca, can I ask you before
Yes.
you leave? I have another actually curiosity question. I noticed
Sure.
you have EC numbers that are not complete in this. So
Yeah.
like one dot dash dash dash. And so does what is the policy of that? I mean, it's not a simple answer. I know that, but is there does the user have control over how complete EC numbers they want to include in their analysis, or is it okay?
Those are mapped from the EC numbers are mapped from
whatever's in the genome, basically.
Whatever's in the genome, and we also have to do an update of Keg, but this is pretty good though, right?
Even it's amazing, but yes,
it's pretty awesome.
Yeah.
Now so, this, go ahead, question.
Omar, there are other tools when you do the protein family analysis. It gives you more control over exactly what protein families or EC numbers you want to include.
Oh, I'm about to show that too. Not so much EC, but I'm showing you this. We're going to do, the authors also looked at virulence factors. And here they provided, but only in PDF form, of the locus tag and what they called the gene name and the function. And it was these tick and these human or these vertebrate pathogens. And so because it was only in PDF form, I had to do the laborious process of taking those and then I mapped them, but I'm going to show you how you can do that so I'm just going to show you one, and I've also got this loaded as well in the workspace so that you could play with it yourself. So these are the Refseqlocus tags, and I'm going to go back to BV-BRC here, and I'm going to show you a new, one of our oldest services, which is the ID mapper. So I can just put those in here, and then I can, it was the Refseqlocus tag. And what I'm asking is, what do you call these in BV-BRC? Because I want to look specifically at this data. So you click the map, and then it shows me what it's called in BV-BRC. And then I can click all of those. I can create in this time a feature group, LPS synthesis genes, just so I don't have to keep doing this. Let's see, why isn't it valid? Well, I don't care. Oh, because I'm saying it's an existing group. It's a new group. LPS synthesis genes. And then once I have this, I can get the protein families. And I won't take you through that step, but I should do the view here so you can, well, let's see, view, zoom to 200. So I was taking these values for the genus specific family. I'm going to shrink it up a bit. So I mapped it to that so that I could do this next analysis. So keep that in mind. Unfortunately, right now, or fortunately, we have to go back to Patrick. And so this service with the comparative pathway was looking at pathways that are mapped to Keg. You can also do a deep dive into the protein families. And so this tool that we're going to be migrating to BV-BRC is called the Protein Family Sorter. So I click on that, and here are my guys here. We want the 2 ticks and 2 of the vertebrates. So Francisella, my endosymbiont, Persica, this guy, this SCHUS4, which is the non-unique one. I'll just use you this time. And then it was a Francisella novacida, but it was U112. I'm just putting this in to show you how vague you can be in the name and still get it. And you can see it here. And this time, because they're all Francisella, I wanted to use the local families, the genus specific families. So I click on search here. And it pretty quickly returned the results. So across these 4 genomes, there are 2,484 protein families. This is your pan genome. The table here has the protein family ID, the number of proteins in this family, the number of genomes that's found across. And now, of course, now it explains it, mobile element proteins, the minimum amino acid length, the maximum, the mean, and the standard deviation. So this is when it's on either mixed, that is the pan genome. If I were to click on present in all, that's the core genome. That's one that everybody has. So those are often things that you need to report in papers. And then I could even click in on this and absent it all, and then this one present in this, and then I can see what protein families are unique to this particular genome. But right now, I want to look at those LPS genes. So remember that I used the ID mapper to find what the protein families were for those genes in a sort of indirect way. I can put that into the filter. And you could put other things in here, like names, which are not necessarily specific, EC numbers, which would probably be okay, but I think protein family IDs work best. And then I click filter. And now it's taken all those 2,000 results and it's shown me 34 instead. So I can see a lot of information about that, but I can get a visual view of it by looking at the heat map. So I click on that, and I can even flip the axis and show this was the order that it was in the diagram. And then I could even order these so that you can see they're a little different. If it's black, it means it's present, and if it's gray, it means it's a pseudogene. For us, if it's black, it means it's absent, which there could be a number of reasons for this. If it's this yellow color, it means there's 1 copy. Or this tangerine color, it means there's 2, and the orange color, it means there's 3 or more. So I want to quickly, because I know you guys have had to sit through this thing, I want to quickly go back to the slides to do a quick summary of this. So could you do similar analysis in the BV-BRC? So let's look at the result comparison. The authors used FastQC, which we couldn't use because they didn't give us the reads. They used spades as their assembly. We have spades. They used DaySys for their annotation. We use the RAS toolkit because it gives us protein families and a number of other things. They created their protein families manually. We use the RAS toolkit to do that. Their phylogenetic tree was using RAXML, which we also had. For genome alignment, they did MAUV. We did progressive MAUV. Pathway comparison, they did that in a manual sense. We have the comparative pathway tool. And for the virulence factor comparison, they had a manual approach to that. We used the protein family sorter. The phylogenetic trees looked similar. This was the alignment of the tick genomes. Theirs is at the top. Remember using MAUV, which I also like, right? I like MAUV, where you can see things that are missing, but we have progressive MAUV. And they did that with the tick, the human pathogen or the vertebrate pathogen versus the tick symbiont, which we did the same here. The biotin B7 pathway, I ordered the genomes in the same order, and then I looked via the EC number at this. Luckily for these guys, they also gave GenBank accession numbers just for this particular group. So I was able to order them in the same way. Now we had a little bit of different results here. They said that the Coxiella symbiont had this. I actually did a blast against the genome of this organism. It really was missing, and this Francisella persica actually had 2 complete copies of it. So you can also do the blast here to see these things. And then also there's the Keg Pathway Map that's provided. And I went ahead and circled the genes that they were looking at in their diagram here, so that they were looking at this particular part of this Keg map. And then finally, for the LPS, the virulence factors, I concentrated just on the LPS synthesis and ordered them the same way. And so you can see sort of similar results, mainly that these guys had basically everything as these guys did. And I didn't do a complete deep dive into what was going on here, but it's something I certainly could. You can see they have a lot of pseudogenes mentioned as present and absent, and some things that are completely missing, which we had as well. So basically, I think when you go back to this question of, can you do similar analyses in the BV-BRC? In this case, yes, I think that definitely you can. And I know I've gone a little long, but I'm open for questions if anybody has any.
It was great, Rebecca. It was a lot of information, but very, very helpful. Yeah, so if people have questions, I'm sure Rebecca's happy to hang on for a few more minutes. Otherwise, feel free to unmute yourself and ask the question, or type it in the chat box. As a reminder, this webinar is recorded and will be shared with everybody, including by email, but also on the website page, on the webinar page. So you'll have access to it, and you could go through some of the tools that Rebecca described.
Yeah.
So I'll be quiet for a bit. Yeah, go ahead.
We have tutorials and everything else, but if you have questions, I'm happy to answer anything. And I know that there's been a lot of information, but you're probably exhausted. Have you guys been monitoring the chat? Are there questions that need to be answered from that?
Oh, I think we took care of them. Yeah.
Okay.
That's great. Okay, I don't think there are any questions. So thanks, everybody. We'll see you soon. Remember, we have additional TIC webinars on the schedule that Rebecca mentioned earlier. So come to our TIC webinar page and make sure you register for them. There'll be something to learn from all of them. And of course, a lot of tutorials and learning materials all over our sites. So definitely take advantage of them.
The ones that are going to be done by me, specifically the bacterial ones, like the metagenomic analysis of tick samples, basically my format will be finding a paper and showing you how you can do the similar analysis in the BV-BRC. So I'll be doing metagenomic. I believe, Omar, you're doing the eukaryotic pathogens? Or...
Yes, I'll cover Babesia and Theileria resources in Pyroplasma DB. Yep.
And I'll be doing comparative genomics of Borrelia next. Anna will be doing Flaviviridae and ticks. And then I'll finish up with RNA-seq and SNP analysis of bacteria transmitted by ticks on June 14th.
Great. See, any last parting thoughts or questions?
I think we can register this one as in the bag.
Great. Okay, well, thanks, everybody. Thanks, Rebecca. And I'll chat with you later about the recording and everything.
Okay, thanks so much.
See you all later. Yeah, bye-bye.
Bye.